Continuation of the previous video. Flower development is a complex process that is tightly regulated by a network of genes and hormones. Then environmental and developmental cues such as light, temperature, and photoperiod play important roles in initiating flower development. The patterning of floral organs is regulated by a complex network of genes that interact with each other to form specific floral structures. Hormones such as auxins, cytokines, gibberellins, and abscisic acid play important roles in regulating floral development growth. Once fully matured, flowers are ready for pollination and reproduction, resulting in the formation of fruits and seeds. Flower development varies widely among different plant species, but the process generally follows the same basic stages. For example, in Arabidopsis thaliana, a small flowering plant commonly used as a model organism in plant research, flower development involves the sequential formation of sepals, petals, stamens, and carpels. In maize, flower development is more complex, with separate male and female inflorescence developing on different parts of the plant. Then, flower development is crucial for the reproduction and survival of flowering plants. By producing flowers, plants are able to attract pollinators and ensure the fertilization of seeds, which is necessary for the production of future generations. In addition, flowers are an important source of food and shelter for many animals, making them an integral part of many ecosystems. Understanding the molecular and genetic mechanisms that underlie flower development is therefore crucial for improving crop yields, conserving endangered plant species, and maintaining healthy ecosystems. Next, plant physiology. Photosynthesis. Photosynthesis is the process by which plants use sunlight to convert carbon dioxide and water into glucose and oxygen. It occurs in the chloroplasts of plant cells and is divided into two stages: light-dependent reactions and the light-independent reactions. The major steps in the process of photosynthesis are light-dependent reactions like chlorophyll pigments absorb sunlight and convert it into energy in the form of ATP and NADPH. Water molecules are also broken down, releasing oxygen as a byproduct. Then light-independent reactions. Carbon dioxide is fixed into glucose using the energy stored in ATP and NADPH produced in the light-dependent reactions. Then respiration. Respiration is the process by which cells break down glucose to release energy in the form of ATP. It occurs in the mitochondria of plant cells and is divided into three stages like glycolysis, Krebs cycle, and oxidative phosphorylation. The major steps in the process of respiration are glycolysis. Glucose is broken down into two molecules of pyruvate, producing a small amount of ATP. Krebs cycle. Pyruvate is broken down into carbon dioxide, producing ATP and electron carriers. Then oxidative phosphorylation. The electron carriers produced in the Krebs cycle are used to generate a large amount of ATP. Nitrogen metabolism. Nitrogen is an essential nutrient for plant growth, and plants obtain it from the soil as ammonium or nitrate ions. The major steps in nitrogen metabolism are nitrogen fixation. Some bacteria and cyanobacteria can convert atmospheric nitrogen into atmospheric ammonium ions, which can be taken up by the plants. Nitrate assimilation. Plants can take up nitrate ions from the soil and convert them into amino acids, the building blocks of proteins. Then ammonia assimilation. Plants can also take up ammonium ions from the soil and incorporate them into amino acids. Then carbon fixation. Carbon fixation is a process by which plants convert atmospheric carbon dioxide into organic compounds such as glucose. The major steps in carbon fixation are the enzyme Rubisco catalyze the reaction between carbon dioxide and a 5-carbon sugar called ribulose biphosphate (RBP) to produce two molecules of 3-phosphoglycerate 3-PGA. 3-PGA is then converted into glyceraldehyde 3-phosphate 3G3P, which can be used to synthesize glucose and other organic compounds. Overall, these metabolic cycles are essential for plant growth, development, and survival. They allow plants to obtain nutrients, produce energy, and synthesize the organic compounds needed for various cellular processes. Then animal physiology and immunology. Muscle contraction. Muscle contraction is the complex physiological process that enables animals, including humans, to move and perform other activities that require the generation of mechanical force. Muscle contraction involves the activation of motor neurons that stimulate muscle fibers to produce force by shortening and pulling on tendons. The contraction is powered by the sliding of actin and myosin filaments in muscle cells, which is regulated by a series of biochemical and biophysical events. The process of muscle contraction can be divided into several stages like excitation. A motor neuron releases acetylcholine at the neurotransmitter junction, which depolarizes the muscle cell membrane and triggers an action potential that propagates along the muscle fiber. Then, muscle contraction is a complex biological process involving the coordinated action of various proteins and enzymes that convert chemical energy into mechanical force. It is a fundamental process in the physiology of animals, enabling them to move, generate force, and maintain posture. This process is essential for various biological processes such as locomotion, respiration, and digestion. The process of muscle contraction can be divided into several steps like excitation. A nerve impulse travels down a motor neuron and reaches the neurotransmitter junction where it triggers the release of the neurotransmitter acetylcholine. Then activation, acetylcholine binds to receptors on the muscle cell membrane, triggering the release of calcium ions Ca2+ from the sarcoplasmic reticulum. Then contraction, the calcium ions bind to the regulatory protein troponin, which causes a conformational change in the protein complex, leading to the exposure of the myosin binding sites on the actin filaments. Then cross-bridge cycling. The myosin heads bind to the exposed binding sites on the actin filament, forming cross bridges. The hydrolysis of ATP by the myosin heads provide the energy for the cross bridge movement, causing the actin filament to slide relative to the myosin filament. Then relaxation. The level of intracellular calcium ions decreases, leading to the dissociation of the myosin heads from the actin filaments and the relaxation of the muscle. The major proteins involved in muscle contraction include actin, myosin, troponin, and tropomyosin. Actin and myosin are the main contractile proteins, while troponin and tropomyosin are regulatory proteins that control the interaction between actin and myosin. The importance of muscle contraction in animals is obvious, as it is essential for movement and the maintenance of posture. Without muscle contraction, animals would be unable to perform many physical critical functions such as walking, running, flying, and swimming. Muscle contraction is also important in the respiratory system, where it powers the contraction of the diaphragm and intercostal muscles, enabling breathing. Next, action potential. Action potential refers to the brief but rapid electrical signal that propagates along the membrane of a neuron or muscle cell. It is a crucial component of the nervous system, allowing for communication between neurons and for muscle contraction. The action potential is initiated when a neuron is stimulated by a neurotransmitter, hormone, or sensory input. This causes an influx of positively charged ions such as sodium into the cell, depolarizing the membrane potential. Once the membrane potential reaches the threshold, voltage-gated ion channels open, leading to a rapid influx of sodium ions into the cell and a rapid efflux of potassium ions out of the cell. This change in ion concentration causes a rapid reversal of membrane potential, resulting in the action potential. The action potential has several important features. It is an all or nothing event, meaning that once the threshold is reached, the action potential will occur with the same magnitude regardless of the strength of the stimulus. It is also a self-propagating event, meaning that the action potential will propagate along the entire length of the neuron without decreasing in strength. The speed of propagation is determined by the diameter of the axon and the presence of myelin, a fatty substance that stimulates the axon. Then axon potentials play a critical role in the function of nervous system, allowing for the transmission of information between neurons and from neurons to muscles. They are also important in the regulation of heart rate and the contraction of smooth muscle in the gastrointestinal tract. Examples include muscle contraction, which is necessary for movement and other bodily functions, and their role in the transmission of sensory information from the body to the brain. In addition, many neurological and psychiatric disorders are associated with abnormalities in action potentials, including epilepsy, multiple sclerosis, and depression. Understanding the mechanisms of action potential is therefore very important. Next, cardiac cycle. The cardiac cycle is a series of events that occurs during one complete heartbeat. It involves the contraction and relaxation of the heart muscles to pump blood throughout the body. The following are the steps and major points involved in the cardiac cycle. Atrial systole. The cardiac cycle begins with the atrial systole, which is the contraction of the atria. The atria contract, pushing blood into the ventricles through the open atrioventricular valves. Then atrial diastole. After the atria have emptied their contents into the ventricles, they relax and the AV valves close. This phase is called atrial diastole. Then ventricular systole. The next phase is ventricular systole, where the ventricles contract, causing the pressure within them to increase. This increase in pressure leads to the closure of the semilunar valves, preventing the backflow of blood into the ventricle. Blood is pumped out of the right ventricle into the pulmonary artery and out of the left ventricle into the aorta. Then ventricular diastole. Following ventricular systole, the ventricles relax and enter ventricular diastole. During this phase, the pressure in the ventricle drops and blood begins to flow back towards the ventricle. This causes the semilunar valves to close, preventing backflow. Then cardiac cycle repeats. The cardiac cycle then repeats itself with the atria contracting again to begin another cycle. The cardiac cycle is regulated by the electrical conduction system of the heart, which ensures that the atria and ventricles contract in a coordinated manner. The system includes the synatrial node, which is the heart's natural pacemaker, and the atrioventricular node, which controls the signals between the atria and the ventricles. The cardiac cycle is essential for maintaining the circulation of blood throughout the body, providing the necessary oxygen and nutrients to the organs and tissues. Any disruptions to the cardiac cycle can lead to heart failure, which can be life-threatening. The cardiac cycle can be monitored using an electrocardiogram or ECG to detect any abnormalities in heart function. Then endocrine system and other systems. The endocrine system is a complex network of glands and organs that produce and secrete hormones, which are chemical messengers that regulate many physiological processes in the body. Hormones are released into the bloodstream and travel to target cells, where they bind to specific receptors and initiate a biological response. The endocrine system works closely with other systems in the body, such as the nervous system and immune system, to maintain homeostasis and respond to internal and external stimuli. Major glands and hormones of the endocrine system include pituitary gland, which are located at the base of the brain, produce hormones and regulate growth, reproduction, and metabolism, including growth hormone, thyroid stimulating hormone, and follicle stimulating hormone. Then there are thyroid gland located at the neck region, produce hormones that regulate metabolism, including thyroxine and triiodothyronine. Then adrenal glands located on the top of the kidneys produce hormones that regulate stress responses, including cortisol and adrenaline. Then pancreas region, which is located behind the stomach, produces hormones that regulate blood sugar levels, including insulin and glucagon. Then there are gonads, like ovaries in females and testes in males, produce hormones that regulate reproductive functions, including estrogen, progesterone, and testosterone. The endocrine system works in coordination with other systems in the body, such as the nervous system and immune system. For example, the hypothalamus, a region in the brain that is a part of nervous system, controls the release of hormones from the pituitary gland to secrete regulatory hormones. The immune system also communicates with the endocrine system by producing cytokines, which are hormones that regulate immune response. The endocrine system plays a critical role in maintaining homeostasis in the body by regulating various biophysiological processes such as growth and development, metabolism, and stress response. Hormonal imbalances can lead to a wide range of health problems such as diabetes, thyroid disorders, and infertility. Nervous system, a complex network of neurons that transmit signals between different parts of the body. The nervous system is responsible for controlling and coordinating many physiological processes such as movement, sensation, and thought. Then immune system, a complex network of cells, tissues, and organs that protect the body against infections and diseases. The immune system recognizes and destroys foreign invaders such as bacteria, viruses, and cancer cells while preserving healthy tissues. Then respiratory system, a group of organs including the lungs and airways that work together to exchange oxygen and carbon dioxide between the body and the environment. We know that. Then digestive system, a group of organs including the stomach and intestine that work together to break down and absorb nutrients from food. Then cardiovascular system, a system of blood vessels and the heart that transport oxygen, nutrients, and hormones throughout the body. All of the systems work together to maintain homeostasis and support the overall health and well-being of an organism. Dysfunction in any one of the systems can lead to various health problems and diseases. Immunology, basic AGAB types, HI, CMI, antigen presentation, MHC complement system. So the immune system is responsible for protecting the body from foreign invaders such as pathogens, tumor cells, and tissue grafts. It consists of complex network of cells, tissues, and organs that work together to recognize and eliminate foreign antigens. So when we learn about that, we can see antigens are there. Antigens are molecules that can trigger an immune response. They are usually proteins or carbohydrates that are found on the surface of pathogens, tumor cells, or transplant tissues. When an antigen enters the body, it is recognized by the immune system as foreign and triggers a series of events that ultimately leads to its elimination. Then antibodies. Antibodies are proteins that are produced by the immune system in response to an antigen. They are also known as immunoglobulins and are able to recognize and bind specifically to the antigen that triggered their production. This binding can help in the elimination of the antigen or its removal by other immune cells. There are 5 major types of immunoglobulins like IgA, IgD, IgE, IgG, IgM. Each type has different structures and functions in the immune response. Next, humoral immunity. Humoral immunity refers to the part of the immune response that involves the production of antibodies by B cells. When a B cell encounters an antigen that matches its specific receptor, it is activated to proliferate and differentiate into plasma cells, which are responsible for the production of large amount of antibody. These antibodies then circulate in the blood and lymphatic system where they can recognize and bind to the antigen, leading to its elimination. Then cell-mediated immunity. Cell-mediated immunity involves the activation of T cells in response to an antigen. T cells are responsible for recognizing and eliminating cells that have been infected with a virus or transformed into a tumor cell. They are also help activate B cells and the production of antibodies. Next, helper T cells. Helper T cells are a type of T cell that play a critical role in the immune response. They are responsible for recognizing and binding to antigen-presenting cells or APCs, which are cells that have captured and processed an antigen. Once activated, helper T cells secrete cytokines, which help activate another immune cells such as B cells and cytotoxic T cells. Cytotoxic T cells are responsible for recognizing and killing cells that have been infected with a virus or transformed into a tumor cell. They do this by releasing perforin and granzyme, which cause the target cell to undergo apoptosis. Then antigen presentation. Antigen presentation refers to the process by which an antigen is presented to a T cell. This is typically done by an antigen presenting cell or APC which capture and process the antigen and then displays it to its surface in association with major histocompatibility complex molecules. Major histocompatibility complex molecules are proteins that are found on the surface of cells. They are responsible for presenting antigens to T cells and are essential for the activation of the immune response. Next, complement system. The complement system is a group of proteins that work together to eliminate foreign invaders. It can be activated by antibodies or by the presence of foreign antigens. Once activated, the complement system can cause cell lysis, opsonization, and inflammation, leading to the elimination of the foreign invader. Like hybridoma technology, monoclonal antibodies. Hybridoma technology and monoclonal antibodies are important techniques used in the field of immunology for the production of specific antibodies. Hybridoma technology. Hybridoma technology is a technique used to produce monoclonal antibodies. It involves the fusion of a specific type of antibody producing cell called a B cell with a cancerous cell called a myeloma cell to create a hybrid cell called a hybridoma. The hybridoma cell has the ability to divide infinitely and produces a large number of identical monoclonal antibodies. The basic steps involved in hybridoma technology are immunization. An animal is immunized with a specific antigen to stimulate the production of B cells that produce the desired antibody. Then cell fusion. B cells from the immunized animal are harvested and fused with myeloma cells to form hybridoma cells. Then screening and selection. Hybridoma cells are screened to identify those that produce the desired antibody. The selected hybridoma cells are then cloned to produce a large population of identical cells. Next, antibody production. The selected hybridoma cells are grown in culture to produce large quantities of monoclonal antibodies. Next, monoclonal antibodies are laboratory produced antibodies that are designed to recognize and bind to specific antigens. These antibodies are derived from a single type of immune cell and hence they are identical in structure and function. The production of monoclonal antibodies involve the following steps like immunization. An animal is immunized with a specific antigen to stimulate the production of B cells that produce the desired antibody. Then cell fusion will be happening, like B cells from the immunized animal are harvested and fused with myeloma cells to form hybridoma cells. Then later, screening and selection will be happening, like hybridoma cells are screened to identify those that produce the desired antibody. The selected hybridoma cells are then cloned to produce a large population of identical cells. Next, antibody production. The selected hybridoma cells are grown in culture to produce large quantities of monoclonal antibodies. Like monoclonal antibodies have several applications, including diagnostic purpose. Like monoclonal antibodies are used in diagnostic tests to detect the presence of specific antigens in a patient's blood, urine, or other bodily fluids. It's also helpful in therapeutics, like monoclonal antibodies are used in the treatment of various diseases, including cancer, autoimmune diseases, and infectious diseases. It's also used in research, like monoclonal antibodies are used in research to study the structure and function of antigens and to investigate the immune response to different diseases. Hybridoma technology is a laboratory technique for producing monoclonal antibodies, which are identical copies of a single type of antibody. Monoclonal antibodies have several applications in medical research and diagnostic purpose, as they can specifically target and bind to particular proteins or cells in the body. The hybridoma technology involves the fusion of an antibody producing B cell with a tumor cell to form a hybrid cell called a hybridoma. The hybridoma cell can grow indefinitely in culture and produce large quantities of identical monoclonal antibodies. The basic steps involved in the hybridoma technology are immunization. An animal is immunized with the antigen of interest, which stimulates the production of specific antibodies by B cells. Then harvesting the B cells. The B cells produce the desired antibody and are harvested from the animal's spleen or lymph nodes. Then fusion will be happening. The harvested B cells are fused with a myeloma cell, a type of tumor cell that can divide independently and form hybridoma cells. Then later, there will be selection process happening. The hybridoma cells are selected based on their ability to produce the desired antibody. Later, we will be performing cloning. The selected hybridoma cells are cloned to produce a pure population of identical cells. After that, antibody production is happening, so the cloned hybridoma cells are cultured to produce large quantities of monoclonal antibody. Let's learn some of the applications of monoclonal antibodies, like diagnostic tests. Monoclonal antibodies can be used in diagnostic tests to detect the presence of specific proteins or cells in a sample, such as in pregnancy tests, cancer screenings, and infectious disease tests. And next, therapeutic treatments. Monoclonal antibodies can be used as targeted therapies for cancer and autoimmune diseases, as they can specifically bind to and block the activity of harmful cells or proteins. Next, advantages of hybridoma technology and monoclonal antibodies. High specificity. Monoclonal antibodies can be designed to specifically target a particular protein or cell type, which reduces the risk of off-target effects. High reproducibility. Monoclonal antibodies are highly reproducible and can be produced in target quantities, ensuring consistency in diagnostic tests and therapeutic treatments. Next, versatility will be there. So monoclonal antibodies have a wide range of applications in medical research and diagnostics, making them a versatile tool in the field. Next, disadvantages of hybridoma technology. High cost. The production of monoclonal antibodies can be expensive, which limits their availability for certain applications. Next, limitations and diversity. Monoclonal antibodies can only recognize a single epitope on a target protein, which limits their ability to detect variations in the target protein. Then ethical considerations will be there, like the use of animals and the production of monoclonal antibodies raises ethical concerns, and alternative methods are being developed to address this issue. Next topic we will be learning tomorrow. Thank you for listening.